Welcome to Click for IB Biology. We're looking at the immune system in topic C3.2, Defense Against Disease. And now we've moved on to looking at the adaptive immune system, which, as I pointed out, is a response system which is found in the vertebrates only. Now, that response system takes two forms, and in this particular video, we're going to be looking at the humoral response. There's a PDF of this presentation that's linked down in the description below. And there you'll also find a link to 10 short answer questions. There's 10 of those. And the answers are provided, and you can use that to secure your own learning. So let's take a look at this adaptive immune system with its humoral response of B cells. So as I just mentioned, the humoral response is based on B cells, properly called B lymphocytes, and characteristically what they do is they produce molecules called lymphocytes, and these lymphocytes, these molecules of lymphocytes, flood the circulatory system and the tissues, and they attack the pathogens and bring about their destructions. However, the system is very specific. That is to say that there's one type of antibody per type of pathogen. But the system actually does have memory, and so it learns from that interaction with that particular pathogen and allows you a faster response time in the future. And quickly though, the cytotoxic response system, in contrast to the humoral system, is based on T lymphocytes. These destroy infected cells, virus-infected cells. So if you've had influenza and the influenza virus has infected your tracheal cells, then this is how you recover. Again, they are pathogen specific, and there is memory in the system. So we're going to focus in this video on this particular side of the adaptive immune system. So in the humoral response, the pathogen has on its surface molecules unique to that pathogen, and we refer to these as antigen. Within the circulatory system, within the system rather, of the host organism, and in particular located within what are called lymph nodes, are located a wide variety of different types of B cell or B lymphocytes. Now, the diagram that I've drawn is trying to illustrate that the glycoprotein receptors in the cell membrane, which are known as B cell receptors, that there is great variety. However, B cells of this form are in very low frequency, and they are located within these lymph nodes. So in order for the correct B cell to be selected, they must come into contact with the pathogen, and that presentation and selection process characteristically can take a few days to occur. So you can see in the diagram that with my pathogen molecule here like this, the antigen like this, this is the corresponding complementary B cell receptor. You can see it's actually this cell here, and now it has been selected. So now we're on our way through the first section, the selection section of the humoral response. The next stage that occurs is that the B cell which has been selected by the corresponding pathogen goes through a rapid cloning process to form two different types of cell. They are the so-called plasma cells and the B memory cells. This event takes place in the lymph node, and on the diagram here I've illustrated in human beings the location of some of those lymph nodes. And when this response occurs, there's a large increase in the number of cells, and in the corresponding lymph node we may experience some kind of swelling, which links into the idea that when one is visiting the doctor's surgery, they often examine our neck for swellings as an early indicator or as an indicator that we may have been going through a humoral response to an infection. So our plasma cells are now a large clone of cells, and they will now synthesize the same receptor glycoprotein in huge quantities. And this enters the plasma of the bloodstream and the tissue fluids. So we'll look at the consequences of these antibodies in the bloodstream in a moment. Just to note that now in the lymph nodes and in the general circulation, we now have a large and higher frequency of what we would call B memory cells, and you can see from the way that I'm annotating their glycoprotein receptor that they are very specific to this particular pathogen. The role of antibodies within the blood plasma and within tissue fluid and tissues is to come into contact with the pathogens and bring about a process called opsonization. So the antigen molecules will act effectively like a glue, sticking the pathogen molecules together and causing them to aggregate together, which is known as agglutination. And so you can see that the pathogens are now very concentrated in this location here. The next step, interestingly, is that we have to return to our innate phagocytes of the innate immune system. And with the pathogens now concentrated, highly concentrated into clusters, the phagocyte is able to engulf these into a phagosome and bring about their destruction in a very, very efficient manner. So curiously, the adaptive immune system through the humoral response enhances the effect of the innate phagocytic immune system. The link here is to the adaptive immune system, taking a look at the cytotoxic response. There's a link there to subscribe to the channel, greatly appreciated for that. And the playlist is here for your convenience for the topic of C3.2, Defense Against Disease.